So now let's finish off talking about the electron microscope and the other types of microscopes that exist in the world. So breaking down the electron microscope essentially has just some basic components. So they're illuminated with a beam of electrons rather than light. So first of all, this allows a much higher magnification. 1.5 times 10 to the power 6 times magnification is actually possible with an electron microscope. Now there's two types of electron microscopes, the transmission electron microscope and the scanning electron microscope. So the transmission, here's the key difference. It allows for the viewing of internal structures such as the nucleus, the cell membrane, the cell wall, etc. These internal structures can clearly be viewed using the transmission electron microscope versus the scanning electron microscope, which is more greater at rendering tiny details but only on the surface of the object. So if you were analyzing, let's say, a bacteria cell, you would be able to notice the flagellum and the other organelles that are on the outside, and the cell membrane and other essential components that are located on the outside of the cell. And they could be viewed using the scanning electron microscope. So this is essentially how an electron microscope works. This transmission electron microscope has an electron gun which shoots out through the electronic beam, right? And there's condenser lessons to magnify the object further, and the object rests right there. The electrons which bounce off and the different scattering of bounces is collected on the fluorescent screen or the photographic plate. So when these different electrons bounce, there's shapes that are formed where the electrons were not able to bounce, and this gives you the surface or the structure of the actual object. And the electrons that do manage to make it to the fluorescent screen are recorded and then transferred to a computer or through the viewing eyepiece as observed here. So we can actually see much greater details of the object itself. Now the other type of microscope is the confocal laser scanning microscope. So here's how it works. It makes it possible to study specimens that are too thick to be viewed using a compound microscope, and they also require cross sections to be prepared. So an example of this would be tree bark. And you can't analyze tree bark directly because it's way too thick to fit on the slide. So with the laser scanning microscope, it produces slices that are composed to form a 3D image. And these are some examples of compound microscope with the nerve cell right here. So this is the detail that we would be able to see. Essentially just the areas where the light is illuminated from. So this would be a dark field type illumination. Then with the transmission electron microscope, this is the detail that we're able to see. As you can see, we can observe the internal structures. This would be the nucleus, the cytoplasm, and the minute organelles inside. Now, with the scanning electron microscope, we're able to clearly analyze the outside areas and determine the structure and the formation of the cell. So this nerve cell, as seen through the scanning electron microscope, is much greater at identifying the outside of the cell versus the transmission, which is the inside. And finally, the confocal laser microscope has the most extreme detail, and this is the rendering of images that we would see on Google Images. For the nerve cell, we can actually get a 3D simulation of the image, which is much more accurate than the other three. Finally, there's one last type of microscope. Now this gives you even greater magnifying power than an electron microscope, and it's 2 times 10 to the power of 8 magnification. And with a computer, it allows for the 3D mapping of surfaces. So this is how projects such as the Human Genome Project and other projects were conducted. We actually get a 3D structure of the proteins and the surfaces and the antibodies and etc. All the little biological components of a cell and the human body can actually be studied in much greater detail with the 3D mapping of it involved using the scanning tunneling microscope. That's how we got images of DNA. So selfies of DNA were taken using a scanning, and many other minute details. All right. Peace.